As humans, we're having many effects on the world around us. Indeed, it's hard to find a place on Earth that hasn't been touched in some way, either directly or indirectly. This is particularly true in the current era of globalization. Many scientists are studying the ecological impacts of these introduced organisms on the native species that currently live there. However, it can be very interesting and informative to study the impacts of these introductions on these introduced populations themselves. For example, we might want to know if the introduced populations have the same amount of diversity as the native populations do. To illustrate this point, imagine a population of birds that come in all different colors. If we were to subsample a small number of individuals and move them to a new population, we might not capture all of the colors present in the original population. And as this population expands, they're going to have less color diversity compared to the original. Now, if we happen to sample enough individuals where we had a better chance of sampling all of the original colors, this new population might in fact have the same amount of color diversity and there would be no decrease. In my own research, I am studying a small songbird called the House Finch. The House Finch is native to the western US and Mexico, but in 1940, it was introduced to the East Coast as a consequence of the pet trade when it became illegal to have wild birds as pets. Within about 60 years, eastern house finches expanded from a small number of starting individuals to hundreds of millions and be almost everywhere. Now, what I'm interested in studying is what were the genetic consequences of this introduction. We don't know how many house finches were introduced. There are reports from 20 to 100,000. So, an important question is, does the introduced population show the same amount of genetic diversity as the native population does? To answer this question, I'm using DNA sequencing. If I take a DNA sequence from two individuals and compare them, there are going to be some number of sites that are different. The number of differences over the length of the DNA sequence is a measure of genetic diversity. Now, I'm not just looking at a single sequence, but at thousands. This allows me to understand what is happening all across the house finch genome. My colleagues and I have sampled house finches from throughout both the native and introduced ranges. And what we have found is that in fact, the native population does have higher genetic diversity than the introduced eastern population, 7% higher. Now, why do we care about how much genetic diversity these different house finch populations have? Well, genetic diversity is the raw material that populations use to adapt to new challenges like new environments, new competitors, or new diseases. And so, moving forward with my research, I'm going to be studying how the introduced and native house finches have evolved in response to a new disease. We'll see if this decrease in genetic diversity had any effect on their capability to evolve.